Imagine the most crucial scientific discovery of the 20th century. The secret to life itself, captured in a single, shadowy photograph. This isn't just a thought experiment. It's the true story of Rosalind Franklin, a brilliant chemist whose uncompromising dedication to precision provided the key that unlocked the structure of DNA. Born in London in 1922, an influential Jewish family, Franklin was fiercely intelligent from a young age. In an era when a woman's place was considered to be anywhere but a science lab, she was absolutely determined to build a career in research. The gender barriers were formidable, but her intellect was even more so. After earning her PhD from Cambridge, a feat in itself, she moved to the vibrant scientific community of post-war Paris. It was here, in France, that she truly came into her own. She became a world-class expert in a highly complex technique called X-ray diffraction. Think of it like this. You shoot a beam of X-rays at a crystallized substance, and the way those rays scatter creates a pattern on photographic film. From that pattern, a skilled scientist can deduce the three-dimensional arrangement of the atoms within. Franklin didn't just learn this technique. She mastered it, perfecting it with an artist's touch and an engineer's precision. This period in Paris wasn't just about science, it was about independence. It forged her reputation as a meticulous top-tier crystallographer, preparing her for the high-stakes, pressure cooker project that would both define her life and, tragically, overshadow her legacy. In 1951, Franklin returned to London, taking up a prestigious post at King's College. Her mission, to map the structure of DNA, the mysterious molecule believed to hold the very blueprint of life. The intellectual challenge was immense, but the social environment was even tougher. King's College in the 1950s was a notoriously difficult, old boys' club atmosphere for women scientists. Franklin was an outsider from day one, and she quickly found herself locked in a struggle, particularly with her colleague, Maurice Wilkins. Their relationship was a disaster of miscommunication and clashing personalities. From the start, Wilkins wrongly assumed that Franklin, despite her world-class expertise, was to be his research assistant. Franklin, fiercely independent and rightly proud of her abilities, saw herself as the leader of her own project, on equal footing with Wilkins. This fundamental misunderstanding created a toxic, hostile atmosphere. They barely spoke, communicating instead through frosty notes and third parties. It was a constant battle for respect and intellectual autonomy. Yet, despite this draining conflict, Franklin's scientific brilliance shone through. Her technical skill was simply unparalleled. Where others struggled, she succeeded. She painstakingly refined the process of preparing DNA samples into a pure, crystalline form, a crucial and incredibly difficult first step. She then spent countless hours fine-tuning the X-ray apparatus, meticulously collecting data with a level of clarity no one else had managed to achieve. Her rigorous work soon paid off. She discovered that DNA could exist in two distinct forms, a drier A form and a wetter B form. This was a critical insight, but it was her work on the B form that would lead to a breakthrough. After months of relentless work, in May of 1952, Rosalind Franklin's dedication culminated in one of the single most important images in the history of science. Photograph 51. This wasn't just another blurry pattern on a film. It was stunningly clear, a ghostly X etched in black and white. To a trained crystallographer like Franklin, this X shape was a dead giveaway. It screamed helix. The image provided unambiguous, hard evidence that the DNA molecule was a spiral. Not only that, but the precise spacing of the spots on the photograph allowed her to calculate the helix's dimensions with remarkable accuracy. Franklin, ever the cautious and thorough scientist, was carefully analyzing this data. She was just months away from publishing a definitive paper that would lay out her findings and propose a structure. But she would never get the chance to present her discovery on her own terms. Unbeknownst to her, Maurice Wilkins, her difficult colleague, took a fateful step. He walked into his lab and showed photograph 51 to a visiting researcher from Cambridge, a young American named James Watson. Watson wasn't a crystallographer, but he and his partner, Francis Crick, were frantically trying to build a theoretical model of DNA. When Watson saw photograph 51, he had a eureka moment. He immediately recognized the helical pattern that Franklin's image so clearly revealed. The image, combined with data from Franklin's unpublished research reports, which had also been shared with the Cambridge duo, without her explicit permission, was the final, critical piece of the puzzle. It provided the framework and the exact measurements they needed. Within weeks, Watson and Crick had built their now famous double helix model. In April of 1953, the scientific journal Nature published a series of articles. The first was Watson and Crick's paper, elegantly describing the double helix structure. Tucked away after it, in the same issue, was Franklin's own detailed paper, complete with photograph 51. The placement was subtle but devastating. It made it seem as though her meticulous experimental work was merely a confirmation of Watson and Crick's brilliant theoretical model, rather than the foundational evidence upon which their model was built. The truth was inverted. Disheartened by her experience at King's, Franklin left shortly after. She moved to Birkbeck College, where she led a highly successful research team, doing groundbreaking work on the structure of viruses, including the tobacco mosaic virus and polio. Her career was flourishing, and she was finally in an environment that respected her leadership and intellect. Tragically, this new chapter was cut short. Rosalind Franklin died of ovarian cancer in 1958, at the heartbreakingly young age of 37. She never knew the full extent to which her work, her photograph 51, had been the critical key to the DNA discovery. 4 years later, in 1962, James Watson, Francis Crick, and Maurice Wilkins were jointly awarded the Nobel Prize in Physiology or Medicine for their discovery of the structure of DNA. In their speeches, Franklin's contribution was barely mentioned. Because Nobel Prizes are not awarded posthumously, she was ineligible, forever excluded from the ultimate recognition. The story of Rosalind Franklin is a powerful, poignant testament to the brilliance of a scientist who provided the crucial moment of truth for the 20th century's most iconic discovery. Her struggle for recognition serves as a vital, enduring lesson in scientific ethics and the immense perseverance required of women who dare to change the world. So, what do you think? Have scientific institutions truly learned the ethical lessons from Rosalind Franklin's story? Let us know your thoughts in the comments below. If this story of the genius behind one of history's greatest discoveries fascinated you, hit that like button and subscribe for more deep dives into the figures who unlocked the secrets of our world. Thank you for watching.